Good afternoon, everyone. We are very much thankful for all of you of joining today's session. We are here live straight away and on the occasion of World Digestive Health Day. We are very much pleased to inform all of you that Zandu Imami Group, they are with us in this initiative that we are doing today a webinar with very esteemed panelists across the country, and we have got a very good response in terms of the registration, and we are looking forward for all of you to join this session and get the maximum leverage and information and knowledge. We are very much happy and pleased to introduce Mr. Gulraj Bhatia, President Healthcare from Jandu Imami Group. Welcome, sir, on the panel today. We have Dr. C.K. Katiyar, sir, CEO Technical Healthcare Jandu. Thank you, sir. Thank you for your time. We have Dr. S. Savarkar, sir, former Vice Chancellor, Gujarat University, Jamnagar, former Chairman, Scientific Committee of Pharmacopoeia, Commission of Indian Medicine, Ministry of Ayush, and former Advisor, Ministry of Ayush, Government of India. Welcome, sir. Welcome on this panel today. And we have Professor Subir K. Maulik, sir, former Professor of Pharmacology, AIMS, New Delhi, currently Professor Emeritus at Indian Council of Medical Research, and Chairman, Interdisciplinary Research Committee, Ministry of Ayush. Welcome, Dr. Maulik as well. So we are very much happy to have all of you on this panel today. It will be definitely a really informative session moving forward, and I request all the participants to please note down your query, your questions. As soon as we will proceed with the session, we will note down your queries, and after our panelists will conclude their talk, their presentations, we will take up your questions and queries, and we will discuss with them. So on that note, I again welcome all of you, and I hand over the session to Mr. Gulraj Bhatia for his welcome note for this particular session. Over to you, sir. Thank you.
Dineja ji. And a very warm welcome again to Dr. Savrikar sir and Professor Maulik sir and Dr. Katiyar ji sir. And to all the esteemed guests who have joined us on this live webinar. A very warm welcome to all of you. As you're all aware, as per Ayurveda, poor digestion is the root cause of almost all diseases. And Ayurveda emphasizes that Ahar, Nidra, and Brahmacharya are the tripod and the platform for leading a long and healthy life. And to have the desired effect of good Ahar, one should have very good digestion or very good Agni. But improper functioning of Agni leads to a lot of gastrointestinal issues, metabolic disorders, and also with the last 2 years, as you know, with the corona pandemic, people's lifestyles have got completely disordered. There's been a lot of stress. So we got many consumers and patients complaining to us that their digestive disorders have increased. And as you know, in Ayurveda, the main principle is to restore and strengthen the Agni in the body. And therefore, in order to remain very healthy, good digestion becomes a very critical requirement. And as you're aware, Jandu has been the trusted Ayurvedic health expert for crores of consumers for over 100 years now, with strong institutional expertise and scientific knowledge and experience of developing products for a wide variety of healthcare needs of consumers. And on the occasion of World Digestive Day, which falls tomorrow, we thought we'll seek the support of the expert panel, which is present with us now, to help communicate to our esteemed medical fraternity and to our patients and consumers on how, on the occasion of World Digestive Health Day, we should proactively help restore our digestive disorders and become more healthy and active. And Zandu Pancharishta, which was launched more than 50 years ago, has always been committed to encouraging and spreading awareness among people to take charge of their digestive system and overall health. So we've launched a number of programs in the last few days and campaigns, both for doctors and consumers and patients, with health camps happening in many hospitals to actually create and build this awareness. And I would like to now warmly welcome our panelists to share their views on how we can actually help consumers and patients to improve their digestive system. Thank you so much.
Thank you, sir. So we'll start with the first speaker, Dr. Savarkar. Over to you, sir.
Yeah. Good afternoon, everybody. Uh, Good afternoon, Dr. Bhatia sir, Dr. Katiyar, Dr. Gulachi, Chitradeep, and all the team from the Zindu and Imami group. Initially, I thank all of you for including me in this very important session, Bhagni. Dr. Katiyar always asks me to speak on this particular subject. So it's a very important day also. The digestive system, the health of the digestive system. Digestive Day it is known as. Now, my friends, I think this is a recognition of Ayurveda again, I will say, because it is said that Agni is very important in case of Ayurveda. Agni, of course, digestion, whenever we talk about digestion, we talk about Agni in Ayurveda. So Agni Moolam, it is the root cause of everything, root cause of health as well as the disease. It is said that whenever if Agni is there, Shantegno Mriyate. It is said as Shantegno Mriyate. Can you increase this slide? Just wait. Yeah, okay, fine. So it is said Shantegno Mriyate Yukte Chiranjivati Anamayaha. It's very important that life and Agni, they are both inseparable from each other. There is an inseparable association between life and Agni. And that's why if there is extinction of Agni is there, it is said it is a termination of life. If the Agni is healthy, life is healthy. And everything is depending on Agni. The health of Agni, that the health, Agni means the health of the digestive system is very much important to your overall health. And that's why today's topic is the relation of digestive system to the overall health. So unhealthy status of Agni again is responsible almost all the diseases. All the diseases are the Agni is important. So status of Agni and course and management of the disease, this is the path of the disease leading towards normalcy. If a person is diseased, then how to make him healthy? So this path goes through the Agni. Actually, this is a very important topic. Now, this, as I said, life and Agni, they are inseparable association. To talk about Agni within half an hour is itself exercise for me also, or for everybody. Wherever you talk about Agni. Because everything is covered in Agni. There is nothing which is not covered in Agni. So whichever diseases are there, except the traumatic diseases, which you call adventitious diseases, all other diseases are connected to Ayurveda, whether it is a metabolic syndrome or whether the infectious disease, anything, any disease you consider, everything is related to Agni. So to keep yourself Agni or your digestive system healthy means you are keeping yourself healthy. That's why anyway, within a half hour, I will try to cover as much as much topic as possible. Now that's why, in other words, we can say that it's a pivot around which all the factors responsible for maintenance of health and causation of these disease, they revolve around this Agni. Or Agni, in terms of today, we can say it is a digestive faculty. Because I have given it a term as a faculty because it's not a single thing. It's not a single enzyme which can be called as an Agni. Agni will cover each and every enzyme whichever is there. So that's why it is better to refer it as a faculty. It's a digestive faculty as such. Everything comes together. You talk about any transformation in the body. It may be a metabolic transformation or maybe a chemical transformation. Any type of transformation, it is already related to the Agni. That's why it's a digestive faculty. Some people you like to say it as a digestive and metabolic faculty also. Because, of course, digestion is ultimately going to convert into metabolism. So that's why it is also known as digestive and metabolic faculty. So I call it as a digestive faculty. And that's why malfunctioning of this digestive faculty is responsible for all types of disease. You consider any disease, it is related to malfunctioning of this Agni or malfunctioning of the digestive faculty. Now, when we consider about this Agni, what is it usually in terms of for general common people, common language, layman's language? What is Agni? Agni is fire. So people many times say there is a digestive fire. There is fire in your stomach. Is it a fire? No, it's not merely fire. Maybe it is having many functionings of a fire, like a heat is coming from fire. So whenever there is a digestion, the heat is always related. That's why, but simple saying an Agni in your body, the Agni relation to your body or your health is not merely a simple fire. Fire is used to burn something. But Agni, whenever you talk about Agni related to health or our body, it means much more than that. It's a plethora of complicated physiological actions responsible for the growth and development of new organism. There are very complicated physiological actions inside our body. They are continuously going on, 24 hours, 24/7, they are going on. So all these actions are related to Agni. So you can say Agni is fire. It's not like that. It's there are very physiological actions. They are combined together. That's why I have used the term faculty. It's a faculty which is taking part in everywhere up to the cellular level. I will say not only cellular, even intracellular level also. Even at the level of the mitochondria or the cell bodies, whatever they are there, this function of transformation from one part to another part or one chemical entity to another chemical is a continuous process. And wherever such transformation is going on, there the role of Agni starts or Agni is involved. That's why we say Agni cannot be translated as a single enzyme. Agni cannot be translated as a simple size enzyme. It's a total, you can say it's a total mechanism or total, there is a system, total system as such, where a number of factors are involved in the action of this particular Agni. So what are those factors and how do they act? I will take just the gist of all those things because that is not possible in this short time. So how is say? Usually, what is the function of Agni as such? Function of digestive faculty or function of digestive faculty? What is it? Usually it is said that the plants are coming from the earth. Earth gives rise to, earth produces plants. The food is generated from the plants. And it is said that our body or any living body for that one, not only human body, but any living body on the earth, it is produced from the food, which is known as Anna. So this body, our body itself is created or generated or produced from the food itself. So everything is related to earth related to plants, plants to food, and foods are related to body. So our body is just a conversion of plants into our body cells or body parts, you can say. So how does it happen when I say that food gives rise or food produces body parts? How does it happen? It happens through the action of Agni. Now, how does it happen? Let us see. It says the body food is made of 5 mega elements, which called as Mahabhut, Pancha Mahabhut. We say that this universe is made of this Pancha Mahabhut. Our body is also made of Pancha Mahabhut. Our food is also made of Pancha Mahabhut. So there is only conversion of these Pancha Mahabhuts which are existing in the universe into the body parts. That goes on, that takes place, and that the function of the Agni starts over there. So conversion of the universal Pancha Mahabhuts in the Pancha Mahabhuts which are existing in our body, that is the function of Agni. So body parts, usually they are nourished by these elements, these mega elements, because they are having similar characteristics. So similar characteristic parts from outside in the universe, they come in contact with Agni and then they are converted into body parts which are having a similar characteristic. So this is the function of Agni or digestive faculty, which takes nutrients from the universe and they transform into the assembly form inside our body. Now, how does this happen? Let us see. Now we know, as I said, that universe is made of Pancha Mahabhut, 5 mega elements. Our body is also for that matter, any living organism. It is also made of 5 mega elements or Pancha Mahabhut. So we can say that every living organism is a microcosm, which is a small universe, which represents everything outside the body. In the universe is represented inside our body as well. So what does Agni represent inside our body from the universe? It is a representation of energy in the universe. Whatever energy is inside in the universe, that is represented inside our body as Agni. So it is responsible for all metabolic transformations which are happening inside our body. They're apparent in any type of transformation, any type of transformation I'm telling about you. So this is responsible. This is visible in the process of ripening of a mango. In the universe, just I'm giving him. The mango ripens. How does it happen? It is the action of Agni which is located inside that mango. Of course, that is also a living thing. So it is transfers, it is a ripening is a process of transpiration of one chemical to another chemical. So that takes place because the Agni which is located inside that mango also. So all living organisms contain this thing and they are responsible for this particular transformation. So in short, we can say just a broad term, Agni. Usually, whenever we say Agni or digestive faculty, usually we refer to a single Agni which is located inside our stomach. So it is known as Zatara Agni. The Agni located inside the stomach is known as Zatara Agni. So usually duodenum, the lower part of the stomach, it is located. and which initiates the digestive process. That's why it is referred as principal digestive faculty. On this particular principal digestive faculty, which acts as a gastrointestinal level, and all the functions or actions of all other agnis, or for that matter, inside all our inside body, they are totally dependent on this principal digestive faculty. So the other two agnis are Bhutagni and Dhatvagni. Bhutagni, Bhut, as I said, there is the fundamental element, there are mega elements. They have their own Agni, their own habitat faculty. So they are known as Bhutagni. Since there are 5 Mahabhutas, that's why there are 5 Agnis. So all people who are attending this particular session, they are Ayurvedic people. That's why I don't want to say what is Bhutagni and what is Mahabhut and all those things. There is no time for that. So anyway, all these Panchamahabhutas, they are having their own Agnis. So this acts at the intracellular nutrient level. The level of action of all these 3 types of Agni, Jatragni, Bhutagni, and Dhatvagni. That's why there are 7 Dhatus. That's why there are 7 Dhatvagnis are there. Their level of action, point of actions are totally different. This Jatragni, which is located in the gastrointestinal tract, it has its area of action inside the gastrointestinal tract. Whereas Dhatvagni levels at the tissue level, broadly I cannot translate directly Dhatu into tissues, but broadly we can say Dhatus are tissues. So at the tissue level, whenever the action takes place, it takes place through the Dhatvagnis. Whereas at the cellular level, at a microcellular level also, the action of Bhutagni starts. So these are the 3 types of Agnis which are taking part in the digestive process. They convert the universal Bhutas into the Bhutas which are located inside our body. So how does it happen? As I said, principal digestive is Jatragni. It's a root of all other Agnis, all other types of Agnis. It digests the foods, converts them into assimilable form to provide nutrition to the body as a whole and to the Dhatu. That means this principal digestive faculty takes up the food, it digests the food, it converts it inside the body, and it takes up to the Dhatus, and Dhatus, they convert their own food, they take their action, the Dhatu start, they convert their own requirement of the food, and accordingly, they are also nourished. The same thing happens at the cellular level also. How do they do? Let us see. In short, we can say there are different stages of digestion, steps of digestions. Through these steps, the food is converted into body parts, our body parts. How do they? In the first place, the material acquires sweet taste at the production of the frothy kapha. There are 3 steps generally. First phase, they are also known as avastha pakas. So this avastha pakas is the first phase, second phase, and third phase. In the first, when the material is ingested, this ingested material is acted upon by the Jatragni at the first phase, where the material acquires a sweet taste and it is converted into a frothy type of material which is known as kapha. So this is the production of kapha in the first phase, which is having a sweet taste. Whereas in the second phase, the partial digested material acquires a sour taste, which is known as a pitta, which is in semi-solid or liquid form. And in the third phase, which takes place in the large intestine, where the Agni changes its role from digestion to absorption. So this absorbs moisture with the production of formed stool along with bile. That means there the feces is formed, the stools are formed, and they are excreted, there the action. That means at the 3 places, in the first place, the kapha is produced, in the second place, the pitta is produced, and in the third place, the vayu is produced, and the excretory part is removed from the body by the action of bile. So these are the 3 steps, very broadly speaking. These steps are known as the avastha pakas. So this avastha pakas, there is again madhura avastha pak, amla avastha pak, and katu avastha pak test. There are tests. This other part, the part is known as nishtha paka or vipaka. There is one known as vipaka. What is vipaka? Vipaka is the taste of the digestive end product. We can say metabolic product, metabolic end product. It has also taste. Now, these tastes are very much. I'm going to speak about the action of taste in my later part of my speech. So here, this I will be covering in a very short time. So this vipaka, it is a taste of the end product of the digestion. There are 3 types of vipakas: sweet, pungent, and the amla also. But some experts do not accept the amla vipak. So there are 2 types of more important principal vipakas. First is sweet vipak, whereas the other is pungent or katu vipaka. The sweet, sour, and salty tasting material, they are converted into sweet vipak, whereas pungent, bitter, and astringent material, they are converted into pungent vipak. So sweet vipak facilitates easy passage of urine and stool, whereas pungent vipak obstructs smooth passage of urine and stool. Many people speak, how do we know that what is the vipak? When it is digested product, how do you know the vipak? The taste of that particular vipak. That taste is assumed or known by indirect perception, by knowing the action of that material. If the person gets easy passage of stools and urine, that means it has a sweet vipak. Whereas if suppose person is getting some difficulty in passage of urine or stool, it is known as katu vipak, which is supposed to be having a katu vipak material. So these are the very important thing which takes place. They also have their own action. So in short, we can say that how does it happen? What is the mechanism of action of Bhutagni? The first thing which, whenever the food is taken, the action of Zatragni starts. Zatragni stimulates the Bhutagni inside the food, because there is also Bhuta in the food also, as I told you. So those Bhutagni, it has a Bhutagni also, that Bhutagni stimulated. It digests that Bhuta material or the elementary material, and that is digestion related to that part. Now this Bhutagni, as I said, the Parthiv Agni will act on only Parthiv part, Jali Agni will act on only Jali part, like that. So all the 5 Agnis will digest their own part. They are very selective in nature. It's not like that. Just like a receptors, they select only receptors, only attack the material which is accepted by them. They don't accept any other specific material. Just like that, even the action is also. I always relate the action of Dhattvagni and Bhutagni to receptor theory, because they are very selective in action. They will not digest any other. They will not accept any other material. They will accept their only own material. Even the Dhattvagnis are also like that. So this digestion of the related part is then. They have food grade, Parthiv Agni will digest the Parthiv part, Apya Agni will digest, like that. So suitable nutrients are taken up, and then only that part of that is nourished. The Parthiv part of our body will be nourished by the Parthiv part, by the action of the Parthiv Agni. That is the action of the Agni, selective action. So that's why it is said that Yatha swam cha pushnanti dehe dravya guna pratak. That means the part of the body which is very similar to the part of the universe, the characteristics of the material which is existing inside the universe, that it helps to nourish that specific part. Doesn't Jale material will not nourish the Parthiv part like that. So they are very selective in nature. So that's why it's a yatha swam samcha pushtanti devuna. And all this action is stimulated by the action of Zatragni, which is very much important. So Bhutagni located in the body as well as in the food ingredient. They are stimulated by the action of the Zatragni and their own Agni. They are responsible for nourishment of the body parts having similar elemental composition. This is very important, my friends. Now, so today also we know the conventional medicine, we say that proteins are digested by proteases, fats are digested by lipases, carbohydrates by carbohydrates, glucose by nucleus, like that. So they are very selective, and the enzymes are also very selective. Just like that, even the Agnis are also very selective in their action. They will not accept other material. They will accept only their own material and digest like that. So this is what, same thing is regarding actions. So simple thing in simple words, plain words. Ahar rasa, the principal nitrogen fluid is converted into dhatu. The action of that one is start. There again, there are two parts, prasada and kitta part. The part which nourishes them is known as prasada. You can say the clarified part is taken up by them, and the waste part is removed. Even that part or some part is pushed ahead, and the next dhatu will be. There is a serial, time is very short, that's why I will not go into those detail. I will show you by the figure. There is one figure also, I will tell you how. You see, this is the figure. You can see here the action, ahar rasa is taken, it is acted upon, rasa is produced, rasagni acts, then it goes to rakta, raktagni acts, then mausa, like that. The whole circle is there, and ultimately the shukra is produced by the principal ahar rasa. So this is a very complicated procedure. It requires a lot of time to explain all this thing. We don't have a time. It cannot be explained within half an hour. So anyway, the nourishment of body is a very continuous process. Agni in a living body working around the clock, as I told you, in one another form. Status of that Agni, it decides the status of other Agnis as well. In short, I will just come to here. Now, as I told you in the last part, some 5-10 minutes I will take just to tell you about the rasa, very important. Because we say that even the food, maybe food or maybe any other drug material. How does that, what is the mechanism of action? We always talk about rasa panchaka. Now, what is rasa panchaka? That means the action is decided by on the basis of rasa, vidya, vipak, guna, etc. So just I will give an example how relevant it is even today in the conventional medicine part also. We know that conventional medicine considers only 5 rasas or 5 tests. Those are sweet, salty, umami, buttery, bitter, and sour tests. Nowadays, they are also considering the another test, which is known as fat test. This is also considered as the sixth test. So umami is known also as meaty, and fat is also in fact fat is also a sweet test. That is a sixth test. So there are test receptors with TRC. There are 4 monographs of type 1, type 2, type 3, and type 4. The type 1 cells are glial cells which detect salty test, whereas type 2 cells are G protein coupled receptors. There are 2 families, T1 and T1R family and T2R family. Their action is very much important. I will just narrate you how this action takes place. Here you can say here, the sweet test is related to T1 and R2 and T1 R3 family, and whereas bitter test is related to T2R family. So these G protein coupled receptors are there. They are located in we only talk about the chemosynthesis receptors which are located on the tongue. In fact, these receptors are spread all over our body. Even the gut and pancreas, they are also having these test receptors. They are very important. Nowadays, it is being accepted. Just before 10 years, nobody is accepting that there are receptors inside our body. So this can very well explain how the vipak also acts. As we say, vipak, kinchit virena, kinchit rasa, gunena pakena, virena chakinchana. That's what we say. Some drugs act by vipak, some act by rasa, some act by virya, like that. So how does it act? Because the receptors are located inside the gut and the pancreas also, or not only gut and pancreas, even all over the body they are present. Now, because of that, those receptors take part in nutrient sensing, balance of hormones, which are essential for the metabolic process. So activation of these receptors, it triggers release of, particularly sweet receptors I'm talking about. It triggers a release of hormones regulating appetite and satiety and help maintain appropriate glucose level in the bloodstream. Whereas dysfunction of these test receptors in gut and pancreas leads to obesity and diabetes. Remember well, I'm talking about the gut test receptors. I'm not talking about the test receptors which are located on the tongue. They are important. But the most important part is the receptors which are located which are distributed all over our body. They are very important. And because of the stimulation of those receptors, the action takes place is very important. So that's why we always talk about the rasa. Every rasa has a different type of action. Just for example, you can say Madhur Rashari, Ayusha, Chardani, Prasado, Balavarnakara, etc. So many actions are there. Every rasa has a specific. How does it act? People will ask them the tongue receptor, it is only sensed by your tongue. Then how does it act inside your body? Because the receptors are spread throughout our body. There the action of these receptors happen. Even the action in the trachea. I will show you how this. Some examples I will be giving you. See, chemosynthesis and GI tract. Sweet test receptor expressed throughout the body. Nasal epithelium, respiratory system, pancreas, islets, sperms, even sperms also, testes also. EC, enteroendocrine cells, collectively largest endocrine cells. We talk about endocrine glands. The EEC, they are collectively the largest endocrine glands, organs inside our body. Nobody was yet considering this thing. Just before 10 years, nobody was speaking about these things. Now they are considering these things. So they are very important because the rasa is taking place. We always talk about rus. People were just laughing at, so how does the rus attacks? But this is very important, my friends. Now they are accepting that these test receptors, they are having their no action and they are located through distributed all over the body. So function of test receptors, this is short, I've said nutrient sensing, glucose homeostasis, secretion of GI peptides, intestinal mucosa, highly expressive test receptor proteins including T1, R20, TR. Their function as a gut liminal nutrient sensor help to regulate glucose balance and nutrient intake. So it is very important to control the nutrition of our body. So we have to consider these taste receptors which are spread throughout the body. T1, R3 and alpha-gastrin express EEC and breast cells of the stomach. Very important. They decide the stomach intake time. Is very important. They secrete ghrelin. You know, that's how we say. I will tell you in the next slide. That bitter test, we say bitter test is Deepak. Now, what does it happen? What is mean by Deepak action? How does it take place? Because it stimulates the ghrelin. Sweet satisfies, it gives satiety because it depresses the ghrelin. So the action of ghrelin is very important regarding leptin and ghrelin. They are very important. So these taste receptors, they are having this process. Rise in blood glucose and amino acid ghrelin release is suppressed because of the sweet testing. So T1R3 expressed in ghrelin producing cells in stomach. T1R3 involved in adjusting ghrelin release after glucose. So that's why after administration of a bitter test, no suppression of ghrelin release takes place, and that is why we say tikta rasa is deeper injection. And by adding some sweetening agent inside your drug, inside your syrup, if you are killing this bitter test, you will be losing the deeper action. That it will not act as a deeper because the rasa are very important in this action. So while drug formulation, these things need to be taken into consideration. Now another thing is the adipose tissue, very important. In the adipose tissues also, the test receptors are located in the adipose tissues also. T1R2, T1R3, they are responsible for adipogenesis, stimulation of these receptors. They form pre-adipocytes into they convert the adipocytes into adipocytes by activation of T1R2 and T1R3. Whose action it is? It is a sweet testing action, sweet test action. So the drugs, the food which is sweet, taste sweet, it is responsible action. That's why the fat cells increase when you eat sweet. Even your saccharin and all those things, they will also do the same thing. They cannot, there is no sugar substitute, remember very well. This is fooling the people. If you are taking sugar substitute by adding some saccharin or other thing, you are fooling the people, because the ultimate action is because the taste is acting, not the substance. Very important. It is responsible for sperm development. That's why we say the Madhur Ras is Vrisha. How does it act? Sperm development and maturation dependent on transduction of sweet test. Very important. So T1 is the alpha-galactosyl. The onset of spermatogenesis, rhythm of spermatogenetic cycle, and aging of test is important. Whereas T1R3 inhibitors like beta test, they lower the sperm count. That's how he said they are responsible for creating sterility, infertility. That's why beta test is not allowed in Vrisha Dravya. Only sweet test is allowed. So this is very important, my friends. This is not a joke that we are talking about rest and all those things. Unfortunately, conventional medicine people do not understand these things. They are now accepting. I will assure you, you know who has received the Nobel Prize last year. The sensors of hot and cold. What is that? It is virya, my friends. They are now saying that, yeah, there are receptors in our body which can see the hot taste of the hot virya of the capsaicin and the menthol of the cold. Nobel Prize they have received. Whereas we are talking about virya so many years. Shita virya and Ushna virya. Now they are talking about that. So this is oral and extraoral places where the receptors are, taste receptors are located. Very important, my friends. I'll just go through all this. Same thing is regarding beta test receptors. They are located in stomach. They confer protection against industrial material, activation of beta receptors in gut stimulation, secretion of hormones involved in appetite stimulation. That's why TIC3 is supposed to be deepen in action. Very important. They induce release of this ghrelin. That's why they are very important. That's why it is deeper in action. Same thing as TIC3 is said to be Vishagna also, because it acts as an antitoxic agent also, the TIC3. Very important, my friends. Don't forget these important things. The Ayurvedic principles, they are very important. Don't try to copy conventional medicine principles. Try to see where they are applicable. Don't follow them blindly. Lot of things which are not told by you, go to see. And food science, very important, food science. Biochemistry, very important for us. Pharmacology is least important for us. You can follow the pharmacology principles, no issue. But don't don't try to find out a drug inside Ayurveda. There is no drug in Ayurveda. If somebody asks me, what is the drug for a particular disease? I will not say there is no drug for your disease. There is a treatment. Yes, I can treat the disease, but I cannot tell you what is it. People ask me, what is a hypoglycemic agent? I don't have any hypoglycemic. I'm sorry. So don't try to do that, my friend. Try to see all this. A very important topic today, Gagni. I congratulate the Jandu team who have selected this particular topic. It's very important. By this thing, at least people will start thinking about the digestive and Agni and all those things. Very important, my friends. So time is very short. That's why I'm not able to explain all these things. It's a very direct topic. Physiological extra T2R family is related to beta testing, innate immunity. If something we know in the corona, people say that Guru Vachi is very important, very good action. Ashwagandha, Guruchi, they are all having beta test. That's why they are having shown very good action in the corona. Of course, many are having the fake also. I don't want to comment on that. But the principle is of course there, which increases the innate immunity. The beta test is responsible for increasing immunity, epithelial barrier, major component. How does it happen? Because it is responsible for immunity, my friends. It prevents microbe entry into the pathogen colonization beta test. It prevents because of the stimulation of the T2 receptors by speeding up microciliary clearance by increasing ciliary beat frequency or by directly producing anti-organism. So this is very important, my friends. These things need to be understood seriously. So these are the receptors, again, T2R receptors, human ciliary. There are families of these receptors are very important. We have to think about that. T2R38 in apical membrane, cilia, human thymus epithelium. Its activation generates nitric oxide, a potent bactericide. That's why it has a bactericide action. All beta testing material. Why they are having it? Because of this particular thing. This is very important, my friends. Beta testers, potentially potent bronchodilators by action of stimulation of these test receptors, produce airway relaxation, protect against airway obstruction, better than some asthma medicines because of these things. They regulate vascular smoothness, T2Rs and test signaling elements in gastrointestinal stomach smooth spell. Beta testin induce a contraction when applied to the lower concentrations. It's very important. Since this is a very new topic, it will take a weight reduction. Again, same thing. High fat, this is T2R receptor decrease. Again, because of the beta testing. But that's why all the drugs in obesity as well as in the diabetes, they are usually testing beta. I just said that 50% of the medicines are bitter. Bitter medicine is all. Whenever there is a good medicine, it is a bitter medicine. There is a power. जो अच्छा दवाई होता है, जो खट्टा जो कड़वा होता है, वही अच्छा होता है. इवन उपदेश के बारे में, एडवाइस के बारे में, जो कड़वा उपदेश हमेशा अच्छा ही होता है. Just like that, the kadwa medicine is always good, the bitter medicine is always good because of these things. So it has got a base, it's not a vague thing. So these are the extraoral receptors, so many things. The action is also shown in the table. You can see this table everywhere. So this is it. So thank you, my friends, for giving me an opportunity. The time was very short. I know that I may have hastened many of my comments, which may not have been understood by the people. But the only thing is that I want to convey a message that this is a very important topic, and it's not that only भूताग्नि and etc. इसमें कुछ दम नहीं है, ये क्या बोलते रहते हैं आयुर्वेद के लोग, ऐसे मत सोचो. ये बहुत important topic है. If time permits, someday I will definitely explain each and everything very scientifically, very nice. Thank you very much. Thank you once again, the Jandu people, Chitradeep, Dr. Khatiyar, Malik Saab hai, उनको भी Piyush Teneja and Guleja Bhatia sir also. Thank you very much for giving me a point. Good afternoon.
Thank you, sir. Thank you, Dr. Savrikar. Very interesting talk. And you finished exactly on time. So I would request Dr. Maulik to go on to the next topic. Very interesting topic. So Dr. Maulik, over to you, sir.
Yeah. Am I audible?
Yes.
Yes,
We can
yes,
hear you, sir. We can hear you and see the presentation.
Good afternoon, everybody. I am very thankful to the organizers, Zandu and Indian Vaidyas, for inviting me as a panelist for such an important meeting just a day before the World Digestive Day. And my friend, Dr. Katiar, when he, what should I say, asked me, I should say asked me, not requested me. He can ask me to take a lecture, you know, that is our relationship. So I said, Sab hum kya bolenge? So then suddenly it occurred to me that this is a topic which is not very old topic, although technically it is very old, but in the modern science, it has taken shape in the last couple of decades, and it is growing up very fast, and a lot of interest has been generated in this topic. So I said, what about gut microbiota in human health? This is immediately he approved the topic. It is not because I suggested, I know him. He will not suggest anything which is not acceptable. So I have taken up this study. By knowledge, if you ask me, what is your knowledge on this topic, I will be in a shaky ground, because I am basically a cardiovascular person. I'm a cardiovascular pharmacologist. So, but of late, in the later part of my career in All India Institute of Medical Science, I had the opportunity to be associated with some of the projects in one of the DBT institutes located in Delhi NCR, where they're working on effect of association, I cannot say effect, association of microbiota, gut microbiota in some disease conditions. And I have generated some interest. I should not say knowledge. I have some, but whatever I will say today, nothing is not my from own research, and there is no conflict of interest in this topic. All are taken from the published work. Due to paucity of space, I have not given proper references, but you take it from me, they are all from publications. So may I go to the next slide? Sorry, I can do it here. Oh, my cursor is not going there. Can you move it to the next slide, please? Someone from the... Yeah. So human microbiota is a very complex system. We can think that we think that bacteria and other unicellular organisms are outside me, so I can protect myself from the surrounding. It is not that easy. Our system has an ecosystem where a lot of microbes, they stay in our body, and they're highest in densities in our skin and the oral cavity and the gastrointestinal tract, mostly the large gut. And to a certain extent, they're also in our bronchial airways. So, which the places which are most exposed to the environment, they are inhabited by this unicellular organism. So, what is gut microbiota? Our gut has got almost 10 to the power 14 cells, roughly, of 1,000 species. 1,000 species, 10 to the power 14 cells. It is a conservative estimate. Some people say that our gut, whole of our gut has got bacteria and fungi 3 times the number of cells that we have in our body, our own cells. So, because of the diversity of these bacterial genes, there are a lot many bacteria and fungi and even virus and yeast. They make a complex genetic environment which collectively is called gut microbiome. Very often, these two terms, the gut microbiota and gut microbiome, are used interchangeably. But there is a fine line of difference between microbiota and microbiome. Microbiota is the cell, and its genetic material is the microbiome. Next slide, please. Unfortunately, I can't control. So, in the last few decades, there is a lot of advances in the genetic tools and related revolutions in understanding our life and our surroundings. And it has helped us in a great way in knowing the composition and function of this microbiota or microbiomes. In fact, during the period of 2013 to 2017, our 13,000 publications were there in 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 uh, in the whole world, which constitutes about 4/5 of the publications over the last 40 years in this topic. Studies have shown that there is a heritable component to gut microbiota. When a baby is born, it is exposed first to the mother's birth canal, That is the time when it gets some of the microcellular organisms inside the body. That has some contribution to it, but overall, the environmental factors, the diet, the medicines, and of course, certain anthropometric measures also are the determinants of microbiota. And it has been established to a great extent that gut microbiomes are key to many aspects of human health, including immune, metabolic, and neurohumoral traits. It is not that it only affects our gut, but it can also affect distant organs like CNS, which is very complex in nature. Different levels of evidence support the role of microbiota in human health. Different levels of evidence. When I say different levels of evidence, because evidence is very important for establishment of science. There are certain evidence which are anecdotal, and certain evidence which are observational, and certain levels of evidence are from experimental studies. It can be animal studies, and certain level of most powerful evidence we generate from the human studies, which can be uh, which can be, of course, a very difficult thing to carry out. Next. What does the gut microbiota do? The next question is this. The gut microbiota provides essential capacities for the fermentation of non-digestible dietary fibers, because a major chunk of our food is not digested, and that acts as a food for this bacteria. And that goes down the gut, and they are acted upon by these microbes. And most important, products of this metabolism or the digestion of these undigestible or indigestible fibers that we take by the microbiota in our gut, especially the large gut, are these three short chain fatty acids: acetate, propionate, and butyrate. And out of this, butyrate has got the maximum importance. Butyrate is the main energy source for human colonocytes, that the cells of the colon, and can induce apoptosis on the colon cancer cells and can activate intestinal gluconeogenesis. And the next point is very important, that it can stimulate epithelial cells to consume a large amount of oxygen, and which causes a state of hypoxia. So there is a relative, relatively less amount of oxygen in the gut environment, which facilitates the production and sustenance of anaerobic bacteria, those bacteria which thrive in a low oxygen concentration or absolutely no oxygen concentration, preventing gut microbiota dysbiosis. This point is very important because I will refer to this point in a couple of my next slides. So this butyrate causes a condition of the state of hypoxia. So that facilitates anaerobic bacteria to grow, and most of the microbiota in our body in the large gut are anaerobic bacteria. Butyrate and propionate control gut hormones and reduce appetite and food intake. It has been found that higher production of short-chain fatty acids correlates with lower diet-induced obesity and with reduced insulin resistance. These sentences are well established in studies. It is not a just flat statement. Unfortunately, I cannot give all the references in a single slide. Next slide, please. The production, another important point is that one important substance is trimethylamine. Is produced from dietary phosphates. This is produced by gut microbiota. And so it has been found that its levels vary in various different people. And trimethylamine has been positively associated with an increased risk of atherosclerosis and major adverse cardiovascular events. It is a major risk factor for many atherosclerotic cardiovascular diseases. Gut microbiota dysbiosis basically is a condition when there is an imbalance between the helpful or beneficial bacteria in our gut and not beneficial or harmful bacteria. There is a balance between these two types of bacteria. If the level of the helpful bacteria goes down, naturally, for some reason, proven reason, the harmful bacteria go up. And this dysbiosis promotes metabolic complications by various mechanisms. And they cause immune dysregulation, altered energy regulation, altered gut hormone regulation, pro-inflammatory mechanisms, such as lipopolysaccharide endotoxins, and they cross the gut barrier, they cross the gut membrane and enter our system, and they circulate in our body. That's why it has been found that dysbiosis has been associated with systemic chronic diseases. That is the reason for it. It has been found that you have germ-free mice for experimental purposes. It doesn't contain any microorganism in the body. And when they received fecal microbes from obese, they start gaining more weight than mice which didn't receive. That shows that those microbiota which are present in the obese person has the ability to increase body weight. Next slide, please.
Dr. Juneja?
Can I have the next slide? Okay, I will try from my end. Okay, how we can manipulate the gut micro? The next question is this. There are two terms we frequently know. Every one of us knows it: prebiotics and probiotics. Basically, for those who are not very familiar with this term, probiotics are basically live bacteria. Probiotics means like bacteria. It is just like bacteria. They are live bacteria, or they are yeasts that, when administered in a viable form and in adequate amounts, are beneficial to human health, and they're usually added like dahi, yogurts, and taken as food supplements. But prebiotics are not live bacteria. They are basically they are called basically microbiota accessible carbohydrates. Prebiotics are substrates. They are basically some indigestible carbohydrates. They are substrates for the microorganisms or microbiota present in our gut. And these prebiotics are acted upon by these microorganisms to produce many things. As I have told you, short chain fatty acids and all, many other things are produced. There is also a term called symbiotics, which is a mixture of prebiotics and probiotics. So probiotics are basically like bacteria. Prebiotics are complex carbohydrates. Next is that prebiotic foods and dietary fibers. Prebiotics, dietary fibers are edible carbohydrate polymers. They're complex carbohydrates, that are resistant to endogenous digestive systems and thus are neither hydrolyzed nor absorbed in the small intestine. So what happens? It goes down the stream. From small intestine, it reaches the large intestine where the actual microbes are there, the microbiota is there. A subset of this dietary fiber sources is fermentable and they serve as growth substrates for the microbes in the distal bowel, that is the large intestine. And these microbes, they act upon this dietary fiber. And their effects on the gastrointestinal microbiome composition has been shown to be beneficial. They provide potential strategy and they provide potential strategy to enhance useful microbiota. So if we can feed this useful microbiota, which is already in our large gut, with this type of carbohydrates, which we don't digest, but they go downstream, go to the large gut, and they're acted upon by the good microbiota, and they produce certain good substances that will be wonderful. And that is exactly what is happening in our body. Low fiber intake, on the contrary, if you take low fiber, complex carbohydrate, reduces production of small chain fatty acids, leading to production potentially detrimental metabolites. So good metabolites are not produced like short chain fatty acids because there's a scarcity of the food for the microbiota because there's not enough indigestible carbohydrates. So it has been found that in Western diet, which contains low fiber, degrades colon mucosal barrier, causing microbiota encroachment, which results in pathogen susceptibility and inflammation, providing a potential mechanism for the links of Western diet with chronic disease. Those who take low fiber diet, their dysbiosis occurs. That good bacteria cannot grow because good bacteria growth is dependent on these products of this carbohydrate. So the bad bacteria, they thrive. The detrimental effects of high fat diet on penetrability of the mucous layer and metabolic functions could be prevented through dietary administration of probiotics. And it is a study which was done by Inulin. Overall, these findings, together with the role of butyrate, again, this butyrate is coming, in preventing oxygen-induced gut microbiota dysbiosis, provide a strong relationship to enrich dietary fiber consumption to maintain intact mucosal barrier function in the gut. What I say that preventing oxygen-induced gut microbiota, because it has been found that butyrate, I have told you that butyrate causes a relatively hypoxic condition in our gut and which facilitates the growth of anaerobic bacteria. So if there's not enough butyrate, which is produced from this high fiber diet, then this facultative aerobic aerobes will grow, which are bad for our gut. And it is a very complex thing. It is not very easy to say all these things in a few moments. So, in tandem with this today's talk, that it's by the Indian by this, and in Ayurveda we use a lot of medicinal plants as medicine for this traditional system of medicine, and it has been found that plant extract rich in polyphenols as potent modulators of the growth of helpful and pathogenic intestinal they modulate the growth of helpful microbiota, and they don't allow the pathogenic intestinal microorganisms. Medicinal plants and their extract substantial quantities have polyphenols, as metabolically active plant metabolites. Polyphenols are food components to wide range of biological activities. Given their poor absorbability in the digestive tract, their activity towards the human host is said to be typically mediated through interaction with intestinal microbes. So the question is, if they are not absorbed, many of the polyphenols are absorbed very poorly, fact, because that is an issue with making druggability issues. How do you give a drug which is poorly absorbed? So, but it is thought that their poor absorbability is sometimes beneficial for the host, because they reach the large intestine, and they are typically acted upon by these microbes to produce good things. Polyphenol rich plants can influence the growth of beneficial microorganisms. It has been proved experimentally that are typical members of this intestinal microbiota. Their dietary intake has been related to the prevention of some chronic, I mean to say that polyphenols intake has been related to prevention of some chronic and degenerative diseases like inflammatory bowel disorders, cardiovascular diseases, diabetes mellitus, even neurodegenerative disorders. Gut microbiota has been associated with depression. It is very surprising. While going through the cross references, I found which are very, very fascinating. Exact mechanisms of polyphenol beneficial activity still remain poorly defined, and it needs to be examined. Beneficial species belonging to genera like Lactobacillus and S. boulardii are often reported to thrive in the presence of polyphenol. These two bacteria are beneficial bacteria, and they thrive in the presence of polyphenols, where the growth of detrimental species is inhibited. In fact, S. boulardii is an extremely potent probiotic that is effective in preventing antibiotic-induced diarrhea, as well as in protecting the intestine from Clostridium difficile and Cholera toxin. World Gastroenterology Organization recommends this microorganism for the treatment of 9 different intestinal diseases, not one: laxatives, hepatic encephalopathy, allergy in infants, inflammatory bowel disorder, cancer, dyslipidemia, 9. And they're all documented. Now I'm coming to my last couple of slides, and these are the slides I wanted to share with you. Given the knowledge that some gut microbiota is helpful, there has been attempt for a very long time to do what we call fecal microbiota transplantation. What they do is that fecal microbiota transplantation is the accepted terminology used to describe infusion of distal fecal material from a healthy donor. A healthy donor gives his feces or the content of the large intestine to a recipient who is suffering from some intestinal disorders which has not treated or effectively treated by the traditional drug or conventional medicines. Thousands of fecal microbiota transplantation treatments are carried out after endemic of Clostridium difficile. Clostridium difficile is a very, very bad infection, which is a super infection, which is caused by some long-term treatment with some antibiotics, which an epidemic occurred in 2000, initially in Canada, followed by in the United States, Europe, and Australia. And this was due to the emergence of hypervirulent strains, and they are resistant to many drugs and increased toxin production. In this setting, this fecal microbiota transplantation was used, and it was found to be very, very protective, and it is documented. And in this reference, I have not put it in the slide because of one reason. Adolf Hitler used to suffer from irritable bowel disease, and he had a very favorite doctor Dr. Hüller, whom he trusted very much, because he said that this disease is because of bad bacteria in your gut. And way back in 1940s, maybe how many years? 80 years ago, the German doctor knew about it, and healthy German soldiers were asked to donate their feces to their Führer, that is their leader, and he used to take it. And it is there in the media. You can check anywhere. And only problem is this acceptability at the present day. How do you expect a patient to take feces? And if you don't think that it is a feces, but actually you are taking a good soup. Good soup means it is containing a good bacteria which will help you in the long run. You can put it in the capsule and take it. And I think I'm the last slide. Can I sorry. No, I'm not on the last slide. So to conclude, the healthy human body is home to an abundance of bacteria, viruses, and fungi, whatnot, that are acquired in early postnatal life. Immediately after birth, you get it, and you keep on getting it. The bulk of microbiome is in the gut, but distinct microbial communities exist in the skin and mouth, nose, lungs, genital tract, everywhere, wherever there's an exposure to the environment. The soup, this is the term I like to use, the soup of metabolites and signaling molecules produced by the gut microbiota could influence many diseases, not to mention behavioral states. Even behavioral states have been involved in these biases. Understanding the mechanisms of crosstalk between the host and the microbiota should offer a more, in our view of the human health, understanding, and that will contribute to prevent, in fact, treating the spectrum of diseases. And the last slide, this is the slide, very interesting slide from British Medical Journal 2018. You see that there are the green things are good ones and red things are bad ones. I'm not going to the details of it, and it is there in BMJ. You can. This is a very good article as far as microbiota is concerned. You can write down, whoever is interested, BMJ 2018, 361 volume, K2. And the pink one are not so good, not so bad. Thank you very much indeed.
Fantastic, sir. Thank you so much for that interesting talk, Dr. Maulik. Very, very interesting. And I'll now request Dr. Katiyar to take over the speaking podium and then talk on his topic. Thank you, Dr. Katiyar.
So, thank you to Tradeep. It was, I will say, feast to the ear, listening to experts and opinion leaders like Professor Savrikar and Professor Maulik. Now, so I always say that throughout our life we remain students. Today I have learned from Dr. Savrikar that taste receptors are throughout the body. Normally we think that taste receptors are only on the tongue, right? And so the complex subject in a simple manner, how to teach the quality of a good teacher. By the way, Professor Savrikar and Professor Maulik, both of them have shown how a good teacher can explain a complicated subject in a simple manner. And when Dr. Maulik was talking about the probiotic, what does it do? It is fermenting the undigestible material. So the moment he said, what came to my mind is probably, I always say that Asavarishta is the unique contribution of Ayurveda in the field of pharmaceutical dosage forms. Because this does exactly that. What body does inside, we do outside. Fermentation of the hardwood, soft leaves, soft flowers, everything together subjected to fermentation, and that fermentation leads to conversion from products to drugs. That's number one. A small amount of alcohol acts as a carrier molecule, helps absorption of actives in a faster manner, and therefore making the product not only more stable but also more effective. So, with this, I am very thankful to the organizers for giving me the opportunity. It's my pleasure to welcome all the Vaidyas who are participating in the webinar today on World Digestive Health Day, which is tomorrow. As Professor Savrikar already has laid the foundation that Ahar, Nidra, and Brahmacharya are tripod of life, and Ahar is the first one. And going beyond that, Agni, every cell is subject to digestion. Every cell is subject to Agni. So if you look at the current concept, whole eco of digestion gets impacted by external factors also like stress. During our student days, we adopted, our teacher was telling, Hari, Vari and Kari. Hari, Vari and Kari were the common cause of the peptic ulcer, because they were not aware about modern medicine teachers. They were not so much well aware about the Ayurvedic concept. So they are putting in a simple term, Hari, Vari and Kari are the common cause of peptic ulcer. As all of you know, indigestion is the root cause of almost all the diseases. And this subject has beautifully been covered by Professor Savrikar by defining 13 types of Agnis. And not only Agnis, 7 types of rasas also, tastes also, how do they affect the body's physiology at a cellular level also. So therefore, good digestion is the key, not only to remain healthy, but also protection from several diseases. So Swasthasya Swastharakshanam is the first objective of Ayurveda. Taking clue from that, Jhandu created a product called Jhandu Pancharishta almost 50 years ago. Today, I am not talking my discussion on this product is more about the research aspect. We have conducted a 2 years research with CSIR, IIIM Jammu, which was earlier called Regional Research Lab. Now it is called Indian Institute of Integrative Medicine at Jammu, part of CSIR, 2 year. And believe me, it's a very, very exhaustive research. I want to share in brief some of the outcomes of the research, which are true not only to Jhandu Pancharishta, but also other Asvarishtas as well. After the research, we have come to know that say Jhandu Pancharishta contains 35 herbs, which constitute the major ingredients of Ashwagandharist, Dasmolarist, Drakcharist, Kumaryasam and Lohasam. Together, they contain more than 1,700 compounds, chemical compounds. Because Jammu CSIR is a chemical lab, chemistry and biology lab, so they have done a lot of research. Almost 1,700 compounds, out of which 495 compounds are reported for their bioactive activity as hepatoprotectives, chloritic, anti-ulcer, anti-spasmodic, laxative, besides compounds having anti-infective activity, helpful in metabolic diseases, anti-inflammatory, antioxidants, and effective on genitourinary symptoms also. The research confirmed that it is not only phytochemical compounds in Jhandu Pancharishta, but during the process of fermentation, some of the vitamin B complex like B12 and B6 especially also get generated, and other micronutrients also get liberated from the plant material and contribute to the beneficial effects. In the pharmacokinetic state, that was the most interesting part. Out of 495 compounds which are biologically active, we did the pharmacokinetics. We found 2 compounds were available even after 6 hours in the blood. Those 2 compounds were quercetin and piperine. So piperine was found to be present in the blood after long duration, and as per the literature, it is responsible for both quercetin and piperine are responsible for protection from hyperacidity, gastric ulcer, adverse effects of stress, protection from injuries to the liver from chemical, besides protection from kidney damage also, and other multiple benefits. The pharmacological activities conducted on the product confirms their anti-ulcer activity, anti-spasmodic activity, enzyme stimulation, like Professor Savdekar said, enzymes are the major part of the digestive physiology. Enzymes like sucralase, maltase, lactase, amylase, lipase, trypsin, and chymotrypsin got stimulated. It was also confirmed that 35 hertz of Pancharishta, their data on digestive system, they are working right from mouth, stomach, liver, gallbladder, pancreas, small as well as large intestines. And besides digestive activity, the polypharmacological effects went beyond digestive tract and also covered the also had the neuroprotective and anti-stress activities. After that, an observational study was also conducted on the product, which confirmed its efficacy on symptoms of indigestion and in most of the cases, complete elimination of the need of the drugs like omeprazole, ranitidine, Digene, etc. after 4 weeks of regular usage of the product in the particular dose. Zendu Pancharishta is one of the popular R&D based innovative product, and we are happy to participate in World Digestion Day today to contribute to the healthy life through the healthy digestion. And probably, let me also share with you, probably it was the first proprietary Asavarishta. Normally, Asavarishta is a category which normally belongs to the domain of the classical Ayurvedic medicine. Very few people have ventured out and converted that into proprietary medicine. So we are one of them, and Zendu Pancharishta has shown excellent data after research at CSIR and some observation study. And till few years ago, it was one of the top selling product. And keeping true to the Ayurvedic wisdom, we are Ahar, Nidra, and Brahmacharya. So for the better benefits of Ahar, today we are one day earlier, one day celebrating the World Digestive Health Day. And during this, we thought it good to introduce, to listen to the leaders on the Ayurveda subject, Professor Savrikar, who, when he talks on Agni, you look at the passion, the way he talks on Agni. I've listened 3 or 4 times, and I always want to listen in more and more on Agni, because he is so clear. My teachers were telling me Agni in 1973, 1974. But even today, I could not understand Agni completely. So it is so complicated a subject, and Agni is directly related with the digestion. And when Professor Malik, when I was talking to him, he said, I will talk on microbiota. That was I was very happy, because a lot of people have heard about microbiota, but very few people know about microbiota. So one of the most important factors today is one, when Professor Savrikar spoke about that taste receptors throughout the body, throughout the GIT. Dr. Malik said that fermentation of the non-digestible material inside the body by microbiota, these 2 are the root causes. And I'm very happy to today, when I correlate all these with the research conducted at IIIM Jammu, I'm very happy to say that probably our product Pancharishta meeting all those requirements. So with this, we welcome all the Vaidyas participating in the seminar, and happy and healthy digestive day to all of you. We celebrate tomorrow. Thank you very much. I am very thankful to the weather.com team for giving me opportunity. Thank you very much.
Thank you, sir. Thank you, Dr. Katyar,
Thank you.
for that excellent talk. So, Dr. Juneja, are there any questions? We've got about 10 minutes for the
Yeah.
timing to end. So, if there are any questions, we could take them and then we could
Yes,
thank
yes,
you.
we have one question first for Dr. Savarkar. So, sir, the question is, after taking heavy meal, I feel muscle pain of my leg. So, what can be the correlation and how Ayurveda can justify this issue? That after people like the person is taking heavy meal and they are feeling pain. So, that is one question for you, sir.
Sir, is it for me? Hello?
Yes, sir.
I can't get the question. What is it? Please, can you repeat?
Sir, the question is that after taking heavy meal, the person is feeling muscle pain of leg. So maybe it is connected to the digestive system, or they want to ask that is there any connection to that?
Yeah. Yeah, I got the question. See, as already we were discussing this thing, everything is related to digestion. Now, pain in your legs. You are saying that taking heavy meal, you are getting the heavy muscle pain in your legs. It indicates that your digestion is not proper. You know, the vayu is related to pain. Whenever pain comes there, vayu is related. And vayu is generated in the last part, third avastha pak. We know that. The ultimate pak is the katu pak. So katu avastha, that means vayu is generated. And the journey of vayu is obliterated somewhere. And that is why you are getting pain. So you take some Vatanuloman ahar. You have to take the Vatanuloman ahar, which is laxative in nature. It will definitely decrease your pain in your legs after taking it. Second thing is you must be taking heavy meals. The heavy meals, that means don't take, according to the principles of Ayurveda, you have to keep 1/4 of your stomach empty. Don't eat full stomach. So that full stomach will be obliterating the path of Vayu, which will definitely be diverted and give rise to pain. So take light meals, keep 1/4 of your stomach empty, and take laxative or anulomak dravya or anulomak ahar in your diet. Then definitely this problem will be solved. Apart from your some other deficiency you may also see, but this is a major cause. Thank you.
Right, sir. Right. Thank you so much, sir.
Thank you so
We
much.
have another question for Dr. Maulik. I think Dr. Maulik is there. Sir, you are there?
Yeah, I think he's there, but
There
internet
is
looks
some, yeah, there's
internet
some
looks giving some problem.
bandwidth issue. We can move on to the next question, Dr. Juneja. We'll come back to this one if there is one more.
Yeah. So, Dr.
May
Katyar,
I answer that question? If it is gut microbiota and cancer, he was asking about.
yes, sir,
If
please.
you
So the
see
relationship
gut
of the
micro,
question is,
yeah,
yeah, please.
definitely. There is a relation not only with cancer but all metabolic syndromes, even metabolic syndromes also. You see, there are two levels of digestion. The digestion at the level our body itself is digesting, and gut microbiota is also a digestive system. Remember, they have their own digestive system. They digest a material which is not digested by our body. Many of the, for example, cellulose or the complex carbohydrates, they go undigested up to the colon, where these gut microbiota, they digest these things. If they are not digested properly, it is continuous digestive part that is going to cause many problems, particularly if you take dry food. If dry food is fiberless food is taken, what happens? Because if there is no fiber, there is nothing in your stomach to contract and pass out. The stomach is empty, and thereby it contracts more and more. The lumen also comes in. The gastric emptying time is lessened. It hassles the food, and that's why there is more friction, and that is going to cause colon cancer. That's why the low fiber is going to give rise to colon cancer. That is the answer for that. Thank you.
Dr. Malik, you want to add on to something? The question is there any relationship between gut microbiota and cancer?
No, the question is very interesting in the sense that it has been said that it is any relationship. I would prefer the term association between gut microbiota and cancer. So far, the studies have as published association that if you take low fiber diet, the higher incidence of colon cancers. As far as the relationship is concerned, they need to have a control studies that they have to have animal studies and all. So clearly there is no mechanistic relationship. There are some isolated, it was there in my slides. There is some dysbiosis and something which causes less of apoptosis because all the cells are shredding and regenerating. So that mechanism is altered somewhere. That is, so association is there, the relationship, the causality is not very clear.
Right, sir. Right. Thank you. Thank you, sir. Dr. Katiyar, one question is for you. Dr. Nakul, he wants to ask that digestive system action of Pancharistin dosh. I think he want to ask maybe Dr. Nakul, what do you mean to say? Be a dosh vidhi you want to say?
Digestion, please say on digestion system action of Pancharishta.
You want to say
In
dosh vritti, I think. Most
those,
Right,
those,
right.
the, I think, yeah, I think it's talking about those, the. So the question is not very clear, but it always depends on the dosa dhatu and mal are the major parts of airway. So we can never leave those on any drug or non-drug. So like that, even Pancharishta also has own role, and it tries to normalize the dosas. So that's what I can say right now. Since there is no, I will say quantifiable measure to evaluate effect of a product on vata, pitta, kapha other than through the symptoms. So the symptoms tell that yes, it is helping in dosa samanyata.
Sir, I want to ask one more. Yeah, Dr. Katiyar, I want to ask one more question here. As far as Pancharishta is concerned, we all know about this product. However, in our doctor fraternity, we are a bit confused. We are confused like generally people came to us for their treatment like a patient. However, as far as Pancharishta is concerned, we also somehow know that it is a general digestive tonic as well. So please give some explanation or some guidance to all the practitioners that how they can balance this particular product in terms of either prescribing to the patients or if somebody is asking as a healthy individual to regulate their digestive system, so how puncturist can help them. So we are just confused that whether we have to prescribe this to the patients or whether we have to give it to the healthy people or both. So there is some confusion in the fraternity. If you can please clarify on that as well. Thank you.
I will, Dr. Juneja, you have asked the very, very right question. And actually, what happens is that if you have a patient who is having a problem of indigestion, which is reflecting in, say, acidity or some other kind of these symptoms for years together. He has been taking lot of omeprazole for 2 years, 3 years. He has been taking Digene, etc., continuously. And despite managing some of the lifestyle, the problem is still there. At that time, if you add Pancharishta as a co-administered drug at that point of time, you will find after 3 to 4 weeks, you will slowly be able to eliminate use of those drugs and then continue Pancharishta for 4 more weeks. So that what will happen when the system becomes abnormal, it takes some time to normal. So first 4 weeks, it will bring the system to normalcy, and later on 4 more weeks to maintain at that. You can say first is the curative dose, and then you give the maintenance dose. So if somebody is taking and I myself, I am not a physician, I am not practicing, but in my family and friends, I have been recommending it to a lot of people. They have been, so even those people who have been taking those antacids for years together, some of them were able to leave after 2 months. Most of them they were able to leave within 4 months. But is the objective is for the patient to leave the antacids? No, objective is to strengthen digestive system. So I think that purpose gets achieved if you add as add-on therapy to whatever he is taking. Because if you ask somebody stop it and take it, no, that's the wrong approach. You add it, and then over the period of time they'll able to leave it, and system will become normalized. As I told you, swasthasya structuram, that is the best part for where Panchadisha strikes. And yes, if somebody is living normal, even he can take it because it will further strengthen the digestive system of this person. So both
Right.
can take it.
Right, sir. Thank you so much. And I think it was a very informative session. We are towards the conclusion of the session now. And anything, Mr. Chitradeep, you want to add on? We can just proceed with the conclusion.
No, I think I would just like to say thank you very much to all the speakers, Dr. Savarikar, Professor Maulik, and Dr. Katyar. And thank you to you too, Dr. Juneja, for organizing this and associating with us. Zandu Pancharist for hosting this webinar on indianvadyas.com, and hope everybody has a great World Digestive Health Day tomorrow. So thank you so much.
Yeah,
Mr.
thanks. And
Mr. Bhatia, anything that you'd like to say, sir?
Yes, definitely. Chitradeep. Firstly, I would express my sincere thanks and gratitude to Dr. Maulik sir and Dr. Savarikar sir for a very enlightened dissemination of many new things for our audience. It was a great learning, as Dr. Katyar sab said also, for someone who's been so experienced like him also. So many thanks to you, sir, for taking time out from the busy schedules. And I would also like to thank Dr. Katyar for sharing his insights on how the digestive system can be improved by using Pancharishta. And to Dr. Juneja for organizing this very well attended seminar. And to all our esteemed guests and Vaidyas who joined us for this talk. And we look forward to many more such sessions on different topics of interest to everyone. Thank you so much.
right, sir. Right. And thank you, all of you. Just to
Thank
update you, at a time, more than 1,200 doctors were watching us live, and we were live on around 8 platforms on YouTube, LinkedIn, Facebook. So we will share the final data. I think it crossed more than 2,000 today. So thank you so much. And we are receiving so many comments and so many questions right now. But due to the limitation of time, we have to conclude now. And I request all of you to please post your queries. We will forward your queries. You can just write your email ID or contact number. We will forward your queries to the Jandu team, and they will be very much pleased to answer all the queries and all the questions. And anyone needs sample or any further detail about Pancharish, please drop your number or email ID to us. We will connect you to the Jandu team and take it for they will take it forward on here. So I again thank all the speakers, Dr. Savarkar, Dr. Maulik, Dr. Katyar, Mr. Bhatia sir, and Mr. Chitradeep for like allowing us to do this kind of sessions with Jandu, and we are very much excited and looking forward to have many more sessions with your team, because it was a really informative session, and I can see the traction and the footfall and the registrations we got and the quality of knowledge shared on this webinar today. So we are very much thankful to all of you and looking forward to have many more sessions in future as well. Thank you and have a great weekend ahead. Thanks.
Thank you.
Thank
Thank
you.
you.
Thank you.
Thank you.